whole genome sequencing of cancers is an extraordinarily complex and technically demanding exercise. And we wanted to push the limits of the technology by studying the most complex genome we could envisage. And that's why we chose this type of cancer, basal-like breast cancer, because it's known to have a very large number of genomic changes compared to normal tissue.
The breast cancer study that we're reporting in Nature this week was a study that compared at the genomic level, the level of DNA, a patient's tumor, her metastatic disease, and her normal genome. In addition to those 3 samples from the patient, we also studied a mouse model that was taken from her primary tumor before it received any treatment, was grown in an immunocompromised mouse strain, and then was later removed, and also studied alongside of the 3 samples directly taken from the patient.
There's several characteristics of basal-like breast cancer that are also of deep interest, and we hope to find some answers from the sequencing experiment. Firstly, this disease is more common in African Americans. And secondly, these tumors are very aggressive if left untreated. But some of them are very sensitive to chemotherapy. Unfortunately, there's a fairly large group of patients in whom these drugs don't work very well. And so getting some molecular insights into drug response was also a primary motivator for the study we conducted.
We used the next generation sequencing technologies to study the normal genome of this patient. It was taken from white blood cells. We also studied from the same patient the cells of the primary tumor and those taken from the metastatic tumor which presented in the brain.
We detected a very, very large number of mutations in this tumor. The complexity was almost overwhelming, leading to the question of what was really critically important to the tumor's growth and what was just background effects of having abnormalities in DNA repair. And what we found was a perhaps novel way of solving that problem by comparing mutation frequencies in the primary versus the metastasis. Because what we found in the metastasis were many mutations that were present in the primary, but their frequency was highly enriched, suggesting the metastasis needed a certain repertoire of cells present in the primary to grow in the brain.
Many of the mutations that we found actually build a case, if you will, for the metastatic process being part of this very aggressive disease. And certainly that was also supported by the fact that the patient went from having a primary diagnosis to having metastatic cancer in only about 8 months. So I think it's a really interesting subset of cancers to study because we don't understand very well what they might respond to, and beginning to tease apart the mutational profile of these cancers is very important to getting to answers along those lines.
We were selecting a subpopulation of cells that were present in the primary, perhaps at a low frequency, but those cells were responsible for the aggressive subsequent behavior of this tumor, both in the brain and in the mouse, where, interestingly, the tumor was also highly metastatic.
The commonly used treatments for breast cancer largely don't apply to basal subtype breast cancers, so they're typically approached with radiation or other very aggressive treatments. If we can better understand the mutational profile of the tumors, we may be able to now suggest new therapies, for example, that would be very specific for the basal subtype tumors.
In many senses, this was a technical exercise. If you like, it was the first genomic Big Bang. We can actually do this kind of science. So, of course, we need to repeat this experiment multiple times to find the entire repertoire of mutations that drive patient fate.